Okay, so
Okay,
Rodrigo.
so again, we have the pleasure to welcome Gemma Huguet for the first lecture of her course, Introduction to Mathematical Neuroscience. And so please be welcome again, Gemma.
Thank you very much, Rodrigo. So this is, as you said, so this is going to be the third lecture of this mini course. And just a reminder, lectures, we saw a little bit about the membrane biophysics of the neuron. And we saw the classical Hodgkin-Huxley model to describe the dynamics of the voltage of a neuron. So we restricted to a single neuron, and then we studied like properties of this type of models and similar models. And in particular, we look at the excitability property. And we make a connection between the different types of excitability of a neuron and certain bifurcations observed in the underlying dynamical system or system of equations that describes this behavior. Today, we want to jump to from single neuron to networks of neurons, or like in neuronal networks. And so I'm going to present you like some of the ingredients that we need to build a neuronal network. And I'm going to discuss the type of dynamics that emerges from a network. Okay, so this is going to be the topic for today. And for the last lecture, what we're going to see is we're going to see applications of this, or like modeling of some cognitive tasks like decision making and perception. So as I said, that the topic for today and for the next session is going to be about neuronal networks. And when we talk about neuronal networks, so we know that the neurons are not isolated in the brain, so they are they make connections with nearby neurons, and or not necessarily nearby neurons. But so when we build a network, the type of ingredients that we need is to know is like, what is the type of neurons that we have in our network, and how many of neurons of each type. And also we need to know about how these neurons are connected. And in particular, let me tell you that the connections between neurons are called synapses. Okay, so we need to know like how are the connections between neurons. And then we need to know also what is the external input that this network receives. And then once we know a little bit about the biological ingredients of the network, then we need to decide which models we're going to use for the neurons and for the synapses. So I will try to walk you through these ingredients for today. So the first thing I need to tell you is a little bit about the synaptic transmission, or like the connection, how is the connections between neurons, and which are called synapses. So we saw in the previous class that when we say that a neuron is active, we can determine that a neuron is active because there are rapid changes in the voltage called action potentials. And these action potentials are an electrical signal, that originates at the initial segment of the axon, close to the soma, and travels along the axon until it reaches the bottom end. And what happens at the end? Well, at the end of the axon is where the neuron makes connections with the dendritic tree of other neurons. So this neuron that is sending the signal is called the presynaptic neuron, is before the synapses, while the neuron that is going to be the receiver is going to be called the postsynaptic neuron. Okay, so this is a terminology that appears, that is classical in the literature in this field. So now let's look at these connections. So the neurons are not glued together, but there is a space in between. Okay, so and this space, which is called synaptic cleft, is where the transmission takes place, and these transmissions involves chemical ingredients, in the sense that when the action potential reaches the axon terminal, so the bottom end of the axon, what happens is that when this electrical signal arrives here, there are these neurotransmitters at the axon terminal that are released. So these neurotransmitters, when the signal arrives, when the electrical signal are released in this space between neurons, which is called the synaptic cleft, and they bind to neurotransmitter receptors of the postsynaptic cell. When they do this, what happens, this causes in the postsynaptic cell the opening of certain channels. And this causes a current that has the effect of affecting the voltage of the postsynaptic neuron. Depending whether the effect is to promote the depolarization of the postsynaptic neuron, we're going to call this synapses excitatory. But if the effect of the synaptic connection is to prevent the generation of an action potential in the postsynaptic neuron, we are going to call this synapses inhibitory. More specifically, so for chemical synapses that involve these neurotransmitters that are released in the synaptic cleft, when they are excitatory, it means that they promote that the action, to promote the generation of an action potential in the postsynaptic neuron. The neurotransmitter that is involved typically is the glutamate, okay? And this can activate two types of receptors. The AMPA, which gives rise to fast synapses in the sense that the channels in the postsynaptic neuron open really fast, causing this current that is going to affect the voltage of the postsynaptic neuron. Or they can be the receptors, if they bind to an MDA receptors, then these changes are slower. For the inhibitory ones, those inhibitory synapses that are what they cause is that they prevent the generation of an action potential in the postsynaptic neuron. So they try to bring the voltage of the postsynaptic neuron down. And in this case, the neurotransmitter that is involved is this GABA, it's called GABA, which activates two types of receptors. GABA A, which gives rise to fast inhibitory synapses, and GABA B, which gives rise to slower inhibitory synapses. So the first one, the faster, are the orders of like 1 millisecond, well, a few milliseconds, while the slower ones are the order of like 10 times this value, okay, or even more. This is just a little bit technical, but what you need to keep in mind is that there are two types of synapses, inhibitory or excitatory. And the effect is this, to either promote the generation of an action potential in the receiving neuron or prevent the generation of this action potential. These are synapses that involve neurotransmitters, so they are called, they are chemical synapses. There are another type of synapses that I'm just mentioning them here, but I'm not gonna give details on them, which are electrical synapses. In those cases, like there is like it's like if the neurons are sort of glued together, sort of. And in this case, these synapses are called gap junctions. This is just in case you hear this word in some paper or literature or in the literature. Okay, let's see how we model this. So what we need is information about whether when the presynaptic neuron is spiking, or it has an action potential, because every time the presynaptic neuron it has an action potential, it causes an effect on the postsynaptic one. So basically, we need information about the input spike train, or like the spike times of the presynaptic neuron. So if this is the voltage trace of the presynaptic neuron, what we care about is like at what, and this is on the x-axis we have time, what we care about is like at which time there is a spike of the presynaptic neuron. So we care about these times. Because every time there is a spike of the presynaptic neuron, this will cause an effect on the postsynaptic one, in the sense that there will be some channels in the postsynaptic one that will open and close. How do we model this? Well, we saw in the previous lectures that the equation for the voltage is typically modeled is a differential equation that involves here certain currents that are the ionic current responsible of generating the action potential. So what we are going to do when we connect neurons, for the postsynaptic neuron, we are going to add this current here that we're going to call the synaptic current. And this current is going to be 0 when the presynaptic neuron is not spiking. And it will take some value, non-zero value, whenever the presynaptic neuron is spiking. So how we're going to model this current? Well, in the same way as we model the ionic currents to generate for the generation of an action potential using the Ohm's law. So this is going to be a conductance times the voltage. And notice here that this term here, this is the reversal, the synaptic reversal potential. And this is going to determine basically the sign of this term, of this synaptic current. So for excitatory synapses, we're going to choose this, the synaptic reversal potential equal to 0. And then this current is going to be negative. So then with this negative sign, this is going to be positive. So we will have a positive effect on the voltage. So it will cause the voltage to depolarize, to rise, to generate an action potential. However, for the inhibitory ones, this synaptic reversal potential is chosen equal to minus 80 millivolts. So this is going to be mostly positive. And then this current is going to be negative. This current is going to be positive with the negative sign, that this will have the effect of pushing the voltage down. So this is the effect of an inhibitory synapses. Okay. And then there is this synaptic term. So the synaptic, the synaptic conductance, the synaptic conductance is going to have its own dynamics. So it will also evolve in time. And basically, it's going to be a function. So it's going to depend on the spike train of the presynaptic neuron. So what this is going to be, this is going to change whenever there is a spike of the presynaptic neuron. Okay, because then it affects the postsynaptic one. So we model it with this sum here. Basically, this is a parameter. This is a constant that determines the strength of the synapses. So how strong is the effect of one neuron onto the other? So this parameter controls this. And this function f models like how is the effect on the postsynaptic one every time the presynaptic neuron is spiking. Okay, and here we have several choices. There are several options that we can choose and that I want to discuss next. So whenever we have the input spike train of the presynaptic neuron, so these are like in this plot A, we have the spike times of the presynaptic neuron. So one option is to model this function f of t as if the effect, as an instantaneous effect of the synapses. And we model with this Dirac delta. And the idea is that what we do is that every time there is a spike in the presynaptic neuron, we take the voltage of the postsynaptic one and we raise it a certain amplitude, which is given by this parameter GC. Okay, so we take the voltage of the presynaptic, of the postsynaptic neuron. Okay, and we immediately raise it by a certain amount. Okay, delta B. So, and we bring this this voltage either okay, or up or down, and this is going to depend whether the synapses is excitatory or inhibitory. This could happen with a certain delay. We can say, okay, the voltage of the postsynaptic one is going to raise by a certain amount, delta B, but after a time d, which is a delay, okay? Because we may think that there is this transmission delay. This is an option. Another that is shown here in B. Another option is that, okay, let's put a little bit of dynamics. Can you repeat the meaning of f? Well, if f is a function of time, okay, that tells me how the conductance changes every time there is a spike of the presynaptic one, okay? So I need to show how this term here changes every time there is a spike of the presynaptic one. So the first option is like something like which is give rise to a non-smooth system, because what you do is like you take the voltage of the postsynaptic one and you add a certain amount delta of P, okay? Another option is to model it as this synaptic conductance raises by a certain amount with a sudden jump, okay? There is a sudden jump, and then there is an exponential decay. So the synaptic conductance decays with a certain time, with a certain rate given by this time constant tau decay, okay? So then, if we model f of t like this, then this function g sin of t that I showed you here, okay, is gonna, I'm gonna answer the question about the delay after this. Let me first say that this function g sin of t, using this type of modeling, is gonna look like this. So every time there is a spike of the presynaptic neuron, there is a sudden rise in the synaptic conductance, followed by a decay with this time constant tau d, okay? Of course, when we have like two spikes coming very close together, what we have is that these they add up, and then the synaptic conductance raises higher. There was a question about the delay. Let me see the chat. Would we expect the transmission delay in the delay model to be variable or constant? Yeah, it could be. I mean, it depends on how you, depends on what you want to model, how is the biological background. So in some cases, you might think that this delay is fixed because of because the time it takes for this chemical process to take place. And then this is of the order of milliseconds, and I can assume that this is constant for all the network. Or you may say that, well, that this process of this synaptic transmission might depend on certain variables. So it could be both, okay? But for the sake of simplicity, I would say that for the moment it's constant, okay? So you can also make this a little bit more realistic, and instead of like that the raising the synaptic conductance is a sudden jump, you can also put an exponential function for the raising part, okay? And then write this function f of t that is called alpha function in the literature, and it combines like an exponential for the raise and an exponential for the decay, okay? Or we can make it even more realistic and then introduce a new variable that now I call it S, that is gonna control like it's gonna be sort of like of a gating variable that controls the opening and closing of the synaptic channels, okay? And we model the dynamics of this variable, of this gating variable, using also a differential equation, as we did for the gating variables responsible of the opening and closing of the ionic channels, using the same type of law that says that the gate goes from closed to open at a rate alpha, and they go from open to close at a rate beta. And the important thing is that the rate at which the channels go from closed to open depends on the amount of the neurotransmitter released in the synaptic cleft. Okay. So if we call this capital T the neurotransmitter concentration, and we can also make this even put more biology in the background and say, okay, the amount of neurotransmitter released in the synaptic cleft, which depends on the voltage of the presynaptic neuron, because every time there is a spike, so the voltage of the presynaptic neuron raises, this releases neurotransmitter in the synaptic cleft. So we can make this concentration a function of the voltage of the presynaptic neuron. And in this case, we are adding more biological details, in the sense that here we need to keep control, also keep track of like the values of the voltage of the presynaptic neuron. And that in this case, this is for instance an example, but there are many other possibilities, is that the amount of neurotransmitter released is a function of the voltage of the presynaptic neuron and is modeled with this function, which is a sigmoidal function, actually. So this is V of the presynaptic neuron, and this is this function f of V, which is going to give me the amount of neurotransmitter released. And basically it says that when the voltage of the presynaptic neuron raises above a certain threshold theta, then a lot of neurotransmitter is released. This means like voltage of the presynaptic neuron very high. It means that the presynaptic neuron is spiking. Okay, it has this action potential. Well, when it is below a certain threshold theta, what happens is that the total amount is close to zero, because if the presynaptic neuron is not spiking and the voltage is at the resting state, so there is not neurotransmitter released. Okay, so these are like different levels of biological detail that we can put in the synapses. So as you can see here, does it make sense of a constant f a long time? No, because if you put a constant f and you assume that this doesn't change over time, you're assuming that the synaptic currents are always there. Okay, it's not that so what I want is that these currents, to have these currents only if the presynaptic neuron is spiking. If not, the synaptic current must be zero, because the postsynaptic neuron is not affected. So this synaptic current here must be zero if the presynaptic neuron is at the resting state. So I cannot make it constant, because this would mean that this current is constant, so that it wouldn't detect any activation in the presynaptic one. Okay, so I told you about the synaptic conductances, and the other day we saw several models for neurons. In particular, we saw the Hodgkin-Huxley model, which was a 4-dimensional model, a very detailed biophysical model for the generation of an action potential. But as you can see here, sometimes what we only care about is like whether the presynaptic neuron is spiking or not. So we only care about the spike times. And there is a 1-dimensional model, and actually considering like simplified models for single neuron allows that when we put them in a large network, then the dimension of the system modeling the network, we want to be as small as possible. So if we consider like even more reduced models for the dynamics of a single neuron, this would be much more useful when we integrate them in a network. And there is one there is a neuron model that is one-dimensional, and it's called the integrate and fire model, which is very useful when we want to simulate a large network of neurons. This integrate and fire model was introduced by Louis Lapic in 1907, many years before the mechanisms of the generation of an action potential were known, that were specified in the Hodgkin-Huxley model. Okay, but it has this model has the advantage that it simplifies a lot the equation for the voltage. Actually, and the strategy to do that is the following. So the idea behind this model is that we don't need to know exactly. So this is, for instance, in this, if you look at this panel B, in this panel B, we have an action potential in blue. Okay, this is spike of the neuron. And the idea is the following. I don't need to put all the details of the biology that describe like how this action potential is generated. How is the voltage raised, the voltage decay. So I don't need all this information, because I only need is like at what time a spike occurs. And the strategy is the following. So I'm going to keep track of the voltage, and whenever the voltage reaches a certain threshold, I'm going to assume that then a spike will occur. And I don't care about like how this spike is generated and all the ionic currents. I'm just going to record that here there is a spike at this threshold. And since I know that this generates a spike and then goes back to the resting state, so I will immediately bring the voltage down to a reset value. So that's the idea behind the integrate and fire model. So I put a very simple dynamics for the voltage, where basically I eliminated all the ionic channels, all the ionic currents, all the sodium and the potassium, which are responsible of the spike generation. So I don't need these currents. I just keep the leak term that is responsible. So these are the currents that are present when the voltage is at the resting state. Okay. And then, yes, I incorporate the I applied term. Okay. And then for the spike generation, basically what I do is this, is like if the voltage reaches a certain threshold, for instance, in this case, V threshold is set at minus 40 millivolts. So whenever the voltage reaches minus 40 millivolts, I will say, okay, here there is a spike. I don't know, then I don't need the details of how this is generated. Here there is a spike, and I just reset the voltage down to a V reset. Okay. And then I just keep integrating. What is the advantage of this? Well, this is a linear model. Okay, so this is linear in B, and it only depends on the variable B. Of course, it's non-smooth in the sense that here there is a jump discontinuity in the voltage trajectory. But for what I need, it's enough. Notice that this is a linear model, we can write it in dimensionless form. It would have a shape like this, and we can assume that when the voltage is 1, then it is reset to 0. So then notice what happens when we vary this parameter of the applied current. Of course, this has an analytical solution because it's a linear equation and it is very easy to integrate. And when time, so for instance, if I choose a value of the input current that is below 1, what happens is that the trajectories approach exponentially the steady state value I equal to 0.5, no, in this case. It doesn't matter at which initial condition we start, okay? The trajectories approach exponentially the value, the steady state value, V equal to I, okay? This is what happens if the applied current is low, in this case is below 1, and then the steady state value is below the threshold. okay, is below 1. So this is what happens now if we choose a value of I applied, which is above 1. In this case, the voltage, if we consider a continuous, the system, the dynamical system, the one-dimensional equation without the reset, what we have is that the trajectory approaches exponentially the value I equal to 2. Okay. But here I didn't consider that there was a reset rule. So now if I apply the reset rule, what I need to do is, like, whenever the voltage reaches 1, I need to reset it equal to 0 and just keep integrating. And what this is how is the solution for this system when we introduce the reset, because every time the voltage reaches 1, I need to reset it to 0. And consider that every time it reaches 1, there's been a spike. Sometimes this is also plotted with vertical lines, like this. Notice here that we have transitioned from having just a stable equilibrium for values of voltage below the threshold to having a stable periodic orbit when the values of the voltage reach this threshold. Okay, so there's been a bifurcation. Okay, we have transitioned from a stable equilibrium to a stable periodic orbit. This is a little bit of a summary of this leaky integrate and fire model. Is that when we inject an applied current, like the one we have here in black, which is weak, then the voltage approaches this stable equilibrium here. Okay, but if the input that we apply is strong enough, okay, then the voltage raises up to the threshold, and every time it hits the threshold, it is reset. And here what we have is this repetitive firing. Okay, and actually here in this case, what we can see is that whenever the neuron, the leak integrate and fire model starts to show repetitive firing, it starts with frequency close to 0, and it raises as the input increases. Okay, so this is like the type 1 excitability that we saw in the previous day. Let me say that we can simulate also this model with an input signal that has a time dependence. Okay, like for instance, it can be something periodic or it can be something noisy, and then we obtain like the values of the voltage, and again, every time it hits the threshold, it is reset to a reset. And we consider that at this time, T1, there is a spike. And at this other time here, T2, there is a spike. And at the time here, T3, there is a spike. So this integrate and fire model provides us the values of the voltage, of the subthreshold voltages, and also the spike times of the neuron. Okay. Now, there was a question. Would you mind explaining why there are no other terms of the form minus G, B minus E in the last equation? So here in the integrate and fire model, because there are these other terms that we had, for instance, in the for the Hodgkin-Huxley. For the Hodgkin-Huxley, we had these other terms, right? But these conductances are 0 or approximately 0 for values of the voltage close to the resting state, or for values of the voltage close to the resting state. And these currents are non-zero and are activated whenever the voltage starts to rise. And when the voltage starts to rise, then what happens is that the sodium channels open. So this term is now not 0 anymore, and it causes the depolarization. So it causes the generation of the action potential. And this term here then activates later, and it causes the repolarization. It causes to bring down the voltage again. So these terms, these ionic currents, are involved in the generation of an action potential. And now, for this model, we don't care about describing that our system describes the action potential. Actually, I only care to know if there is an action potential or not. And to know that, I only need to, I can remove that and just, I need to control whether the voltage reaches the threshold. Because I assume that whenever it reaches the threshold, it will produce an action potential, okay? And that's the thing, okay. Now, so if we go back now to build our network, okay? So basically, at the beginning, I said that we need to decide how many types of neurons we are going to put. And also, how many neurons of each type, in the sense that we need to decide like how many populations of neurons we are going to have. Like, okay, we consider a population of neurons is neurons of a certain type that they interact, that they have connections with each other. And these populations can be excitatory or can be inhibitory, in the sense that they can excite other cells or they can inhibit other cells. And we can, depending on the type of problem, we're going to have different types of populations that describe like different types of properties of them. And also we need to decide how many neurons we are going to put in each population, like 100, 1000. Also, there are models that they study the limit when n tends to infinite, even if this is not realistic, but you can assume that you have so many neurons that you can assume that n tends to infinite. So this is a decision that when we build a network, we need to know like what type of biology we have in order to build our network. Also, like when we have the neurons, we need to decide like how is the connectivity matrix. That is, we are going to assume that all the neurons are all-to-all connected. That is, each neuron in the population makes connections with all the other neurons. Or we're going to assume that they are randomly connected, with that each neuron is connected with another one in the network with a probability p, for instance. Or we can assume that there is a spatial structure in the sense that each neuron is connected only to nearby neurons, maybe, okay, or neurons at a certain distance. Or maybe these connections might have a structure that is imposed by learning, because when we learn, we modify we create new connections, we destroy other connections, we strengthen some connections, we weaken other connections. So maybe they are modified by learning. So this is another, you can see here that there are many choices that we need to take to build our network. Also, like, okay, this network is receiving also not only the inputs from other neurons in the population, but maybe there is an external input due to like an external stimulus, or like the input from another population, okay? And how is this going to be, this input? Maybe constant, maybe stochastic, maybe it has a structure in the sense that it activates only certain neurons in the population, but not all of them, etc. So here there is also many choices to do. Then when we build our network, we also need to decide which neuron model we're going to use. So for each neuron, we need to describe its voltage and its well, it's dynamics, no? So are we want to use a Hodgkin-Huxley model or Hodgkin-Huxley type of model that describes like the spiking dynamics, or we're going to have a leaky integrate and fire model as the one that I explained. So depending on the choice we're going to do, the overall network system is going to be more or less complicated. And finally, I presented you several options, several models to model the synapses. So we'll need to choose like which synaptic model we're going to use. More or less detailed, more or less simplified. Okay. So based on all these ingredients, you can build like, and you can study different types of networks and different properties. Okay, and also everything depends also on the question you have and the area of the brain you're studying and what you want to understand. In particular, some questions that there exist is like, well, we want to study the dynamics of this network. What type of things we want to see? First is like, what about the spontaneous activity? So when we build our network, we put the model for each neuron, we write the model for the connections, etc. What is the dynamic that emerges? If there is no external input. What happens then? Also, the question about coding. What happens when there is an external input? How does the network encode information about the external input that receives? How this is encoded in the dynamics? Also, questions related to learning and memory. So if this network receives a stimulant and has to memorize this, how does it do? How does it how does the network learns or memorizes an input? And finally, how they do computations. So basically, we need to understand the dynamics of the network. And then, so if we take, for instance, a network with like 10,000 neurons, and each neuron is modeled with a leak, integrate, and fire, and we put them all to all connected and see, like, and then how do we study the dynamics that emerges? Because this is a very huge dimensional system, because for each neuron we have an equation. And how do we do this, or how we visualize the dynamics? So we visualize the dynamics using the raster plot. What is the raster plot? So the raster plot plots on the x-axis plots time, and on the y-axis it plots what it's called the neuron index. So if we label each neuron by neuron number 1, number 2, number 3, so each line here corresponds to a neuron in the network. So for instance, if we take neuron number 1, what we're going to do is, like, as we move along the x-axis, we're going to draw a dot every time this neuron number 1 does a spike. So we keep track of the voltage of this neuron, and every time this neuron spikes, we put a dot. And this is going to be my first row. Then on my second row, on my second row, I'm just going to put a dot every time neuron number 2 spikes. And I'm going to do this for neuron number 1, number 2, number 3, up to n. Okay, which is the total numbers, the total number of neurons that I have in my network. And as you can see, when you see these plots, which puts a dot every time the neuron labeled in the y-axis spikes, you can have like a visual idea of what is the dynamics here. You can see that there are periods where there are a lot of neurons fighting together, followed by periods of like where most of the neurons are silent, and then another period where most of the neurons are fighting together. So this is something that we can visually observe from the raster plot. How do we compute this? Well, what we compute is what it's called the firing rate of the network. Okay. What is the firing rate of the network? Basically, the firing rate of the network takes a time bin here, delta t, and in this time bin, it counts how many spikes are here. And then this is divided by n, which is the total number of neurons, and by this delta t. And we do this for different time bins, okay? And we compute this value and we plot it here, okay? In this function down here. this is the firing rate. It tells us what is the average activity of the network, or in other words, how many spikes per second the network produces. Okay. And actually, with the firing rate, usually indicated by the letter R, what we have an idea is like the overall activity of the network, like what is the, and we can observe, like looking at the firing rate, is this oscillatory behavior, like of periods of high activity, followed by periods of like where most of the neurons are silent. I think there is a question in the chat. Ah, no. Okay. This firing rate variable is something we wanna, is an information we wanna have from the network. Okay. So basically, see, we consider a network of spiking neurons, okay, which describe the activity of a population of neurons coupled through a certain network connectivity matrix. So this means that we have a network or a system which consists of order n coupled differential equations. So there's basically, we need to deal with this. To deal with this, we need to simulate it in a computer. But on the other hand, it is possible to design models or like to provide a set of equations that describe the evolution of the firing rate directly without needing to generate or to simulate this large system of differential equations. So the rate models, also called like firing rate models or mean field models or neural mass models, describe the activity of a whole population by a single average firing rate variable. So basically, what we would like to have is an equation, a differential equation that describes the dynamics of this variable R without need to simulate the full system of differential equations. It is possible to measure experimentally firing. Yeah, well, there are the local field potentials, which in some sense, which are recorded in experiments, and in some sense are related to this, in some sense, yeah? Otherwise, you can always do, like if you have recordings of several neurons, like hundreds of neurons, you can always do this averaging technique, or like this, sorry, this, yeah, or this mean field, or this, that I showed you, this computation that I showed you before. So we have, this is again a raster plot, okay, of two populations of neurons. Like these ones are excitatory neurons, and these ones are inhibitory ones. Okay, so this corresponds to the spike times of the inhibitory neurons, and this raster plot indicates the spike times of excitatory neurons. And we can do this computation of the firing rate, and we have that the firing rate for the inhibitory ones shows this oscillatory behavior, and that the firing rate of the excitatory ones shows this oscillatory behavior here, with a damped oscillations here. As you can see also from the raster plot, the oscillations become less and less intense or marked. And in general, it is not possible to have a differential equation that describes exactly the behavior of a spiking network neuron, of a spiking network. But in some particular cases, there are like in the last 5 years, there's been some contributions where some people in particular, in these works of Montbriot, Patho, and Roxin, that they are capable for networks of leaky, type of integrate and fire models that are called quadratic integrate and fire model, when they are all-to-all coupled, and assuming that n is large, so n tends to infinite, they are capable to produce a set of equations that describe the firing rate activity exactly. And this is what this plot is showing, is that the red trace is just the integration of a differential equation with just a few equations. While the black that you see here in the background is the one obtained from the simulating the full network and just computing the firing rate from this full simulation. But in general, this is not possible. However, there are some models, firing rate models, that have been used classically and describe the average firing rate of a network in a, let's say, qualitative way, in the sense that we are going to have an agreement, a quantitative agreement between the simulation of the firing rate and the simulation of the spiking neuron, of the spiking network. But qualitatively, these firing rate models kind of generate the same type of dynamics. And one of the most well-known model that describes the average firing rate are the Wilson-Cohen equations that they were proposed in 1972 by Wilson and Cohen. And they appear a lot in many papers in the literature, in the field of mathematical neuroscience, because they have become like classical as the Hodgkin-Huxley model. And then describe the average of activity of a network consisting of a population of excitatory cells and a population of inhibitory cells, for which the excitatory cells excite themselves and also excite the population of inhibitory cells. And the inhibitory cells also inhibit themselves and inhibit the other population, the excitatory one. And apart from this, these two populations may receive an external input due to other sources, okay? And this is modeled using this system of differential equations. So we have, notice that this is two-dimensional. We have an equation for the firing rate of the excitatory population and an equation for the firing rate of the inhibitory one. And the shape is like this. This function S here is again one of these sigmoidal functions of logistic functions. So that they look like this, okay? They go from 0 to 1, and there is a threshold theta at which they take the value 1/2, exactly, okay? What is the idea behind? Is like, we consider all the inputs that the excitatory population is receiving. Okay, what are the inputs of the excitatory population receives? Well, it receives excitatory input from itself. This is given by this term here, and is proportional to the firing rate activity of the excitatory population. So the higher the activity of the excitatory population, the more excitation it sends to itself. And JEE is a constant that is a parameter that determines how strong are these connections. Okay, so this is a parameter. What else? Which other input receives this population? Well, it receives inhibition from the inhibitory population. Since it's inhibitory, we put this negative sign here. And how strong is this input? Well, it's proportional to the firing rate of activity of the inhibitory one. So the higher is the more firing of the inhibitory population, the more inhibition sends to the excitatory population. Okay, so it's proportional to the firing rate of the inhibitory population. And again, this is a parameter, okay, and it controls how strong is this connection. And finally is the external input, okay, that might be constant, it might be time dependent. So here there are many ways to play with this. So this is the total input that this excitatory population receives. If this total input exceeds, so look down here, exceeds a certain threshold theta, so this total input that, let me call it X. So if this total input X is above a certain threshold theta, this S is close to 1, And this, the excitatory firing rate tends to 1. Assume that 1 is the maximum, okay? So it's normalized. While if the total input that this population receives is below the threshold theta, what happens is that this value is close to 0, and then the activity of the excitatory population gets close to 0, okay? The same equation is used to model the activity of the inhibitory population, okay? And both of them, they have this constant tau here that determines like the time constant of these populations. So what is the, and with this, I finish. So what is the dynamics that emerges here? Of course, here we have several parameters. Assume that the connectivity is fixed, okay? And that we vary the external input to the excitatory population and the external input to the inhibitory one. So this is the external input to the excitatory population, and we vary the external input to the inhibitory one, okay? So we draw like what is called a bifurcation diagram, a two-parameter bifurcation diagram. So we look at the type of invariant objects and bifurcations of these invariant objects when we vary both parameters, okay? The other ones are fixed. And interestingly, what we find is a region here in yellow where this network shows oscillations, okay? And what does it mean, oscillation? So that and what generates these oscillations? So the excitatory cells, if they receive enough strong input from the outside, they activate. And they activate via the connections, the inhibitory ones. But at its turn, the inhibitory ones suppress the send inhibition to the excitatory ones, suppress the excitatory ones, and then since they receive less input from the excitatory because they are suppressed, they also, its activity also goes down. And then the cycle restarts again, okay? These bifurcations in this Wilson-Cowan model, they might appear through a half bifurcation, if you remember what we saw the other day, in which we transition from having one stable fixed point, okay, so one stable, which is focus type, that is stable, and then this focus becomes unstable at some point, and this is a supercritical half, and what appears there is a periodic orbit, a stable periodic orbit, and this is what we are showing here. So this is in color correspond to the two null planes, okay? Remember that the null planes correspond to the values at which the velocity vanishes, and when they intersect, this corresponds to the equilibrium points of the system. So what we have is that in this pink dot here, if we choose the parameters in this pink dot, what we have is an unstable focus surrounded by this stable periodic orbit. And this periodic orbit, we look at the evolution of the firing rate of the excitatory population and the firing rate of the inhibitory one over time, we see this oscillatory behavior. In a similar way, we can have oscillations that are born through a saddle node on an invariant circle bifurcation, a SNC bifurcation, okay? And this happens when there is a saddle and a node that coalesce, and at this moment, we have a homoclinic orbit, and this homoclinic orbit persists and remains as a periodic orbit after the bifurcation, okay? And in this case, this orbit inherits the dynamics of the homoclinic one, and it has a sort of a slow fast behavior in the sense that it spends a lot of time in this region here, while this is done very fast, this other circle. And this is shown in the picture down here, okay? Then with these Wilson-Cowan equations, I stop for today, and just let me say that in the next lesson, we are gonna see models of decision-making and perceptual and visual perception. Okay, so we're gonna see a practical application of this in in a cognitive task. And finally, I leave here a set of references. And I'm going to stop now because I think it's time.
Okay.
And I
Thank you. Thank you very much. So I stop recording.